Without further ado, my very great pleasure to introduce our first speaker. Dr. Lucy Wedderburn is going to be talking to us about indispensable tools, leveraging biopsy samples for enhanced JM diagnosis and treatment plans. Dr. Wedderburn is a professor in pediatric rheumatology at University College London and the Great Ormond Street Institute of Child Health. She acts as a consultant at the Great Ormond Street Hospital. She trained in immunology and rheumatology in Cambridge and then London, then spent time training in science at the University of Stanford in the USA, before returning to University College London and the Great Ormond Street Hospital on a Wellcome Trust Fellowship. She's been a consultant at the Great Ormond Street Hospital for more than 20 years. She runs a research team that investigates the mechanisms and different types of childhood myositis and arthritis. As well as that, she's the chief investigator of a large cohort study in JDM that includes centres from across the UK, is the director of the newly established Arthritis Research UK Centre for Adolescent Rheumatology at UCL. And she is a key member of our medical advisory board. And I would invite Dr. Wedderburn to talk to us today about leveraging biopsy samples for enhanced JM diagnosis and treatment. Over to you, Lucy.
Thank you very much, Andrew. Can you hear me okay?
Perfect.
Yes.
The slides are perfect.
Okay. If they don't move or they don't appear to be working, please just shout, because I can no longer see you or the chat bar. Okay, so thank you very much for that kind introduction. And thank you to CureJM for asking me to talk to you today. I'm really delighted to sort of join you on this meeting. It looks like an amazing lineup after me. And it's great to see how much interaction there is between clinical thoughts and research across these CureJM events. So I'm going to start just by showing you my disclosures, and I always put our grant funding up front, and I will have further acknowledgements at the end of this talk, but there's one rather unusual disclosure I need to share with you, which is this individual who's actually on the call, so if there are very tricky questions, we'll bring Meredith in. So some of the unpublished work that I'm going to show you at the end is led by a program of work that Meredith leads that's now involving muscle tissue as well as the blood of our patients to really understand mechanisms and also novel targets of treatment. So I guess, as Andrew said, I've been in this space for quite some time now, and we have the huge privilege, really, of working with a UK-wide network of investigators, but not only clinicians and investigators, but also really good collaborators. And one of the things we did early on was to start to look at the actual site, or one of the sites, of the damage to the body that's going on in JM, and that, of course, is the muscle. And like many others, we found a clear signature that of change in that muscle, as well as the fact that it's weak, but also in its biology, indicated by molecules that are driven by this cytokine we call interferon. And it's taken us this long, so 20 years really, to finally get to the point of being connected enough internationally, but also to bring companies on board to the idea to test blocking interferon. There are a range of molecules we could use to do that. And I'm not going to talk about those today, but some of you will be aware we're very excited about those new trials. So way back, actually, some of this data was from Lauren Packman's lab. And then we did some data initially, at some studies initially looking at molecules in the muscle that are driven by this cytokine called interferon. One is a routine stain that we do anyway in muscle analysis called the MHC class 1, and also this protein called MXA, some of us call it MX1 in other analyses. And that was clearly different from healthy control muscle, which we can occasionally obtain in Great Ormond Street through children having biopsies when they're actually otherwise well. And so we can compare this in JDM. And interestingly enough, the amount of this protein that you see on the muscle was directly correlated with how weak the child is. Now, that doesn't mean that interferon is causing the weakness directly. It may have other things in route to doing that, but certainly the two were correlated. And this analysis, done by Claire Deakin and a very talented clinical fellow that we had working with Claire, had a look across the antibodies. And in fact, the different myositis-specific antibodies show slightly different levels of this interferon-driven signature in the muscle. At Great Ormond Street, way back in 1999, we made the team decision that every new child with juvenile myositis would have a biopsy. That's rather an unusual position, especially nowadays, and a lot of people questioned why we did that. I'm now extremely grateful that we did make that decision because I think we've learned a lot. We've also been able to inform clinical decisions, and we have an incredibly valuable resource which families and patients are happy to help us by donating towards, to actually really interrogate the pathology of this disease. And some years ago, actually in about 2005, so 20 years ago, we thought we'd better really bring together some experts in the field of the pathology of this disease, because we have this incredibly precious resource where, because all the patients are kind enough to give us a tiny donation of muscle, we can assess muscle pathology and mechanisms from the whole range of JM. And you all know, living with the experience of JM, how different cases are. They're not all the same, and indeed they're not all the same in their muscle. But if you're in a hospital that only takes a biopsy when things aren't going so well, and you're 2 or 3 years into steroids, and actually the diagnosis becomes perhaps questioned, then those cases won't really serve the purpose of what we wanted to do, which was assess across the spectrum of juvenile myositis. And these wonderful people came from around the world. Every single one accepted our invitation. And I just want to highlight here a colleague who sadly we lost this year, Hemlata Bassani, who very much drove the organization of these meetings. She really was a complete powerhouse of organization. We couldn't have done it without her. And essentially what we did was we used our first 33 biopsies in the first part of this study to ask them to identify and, by consensus process, agree upon a series of fairly easy to recognize features that they felt they could score, that could be easily done in any routine hospital that has immunohistochemistry, which most hospitals have these days. And they came up with 4 so-called domains. These are just areas of the, or topics of the difference that we see in juvenile myositis muscle compared to healthy muscle. And it had to be age-matched muscle because muscle changes as we get older. So the fibers get bigger and they're different in boys and girls. So we really needed to look at control biopsies as well. And they ended up with these 4 domains. The score tool is published, as are the very detailed instructions that they generated. And here are those instructions. They fit on one piece of paper. So the idea is that really any laboratory in the world could do this. This is not a research tool. It's a tool to be used in the clinic. It's not a diagnostic tool, but I think sometimes actually it's incredibly useful in the diagnosis. Also, we have a sense of severity and which aspects are more severe or less severe. So we're now able to compare children across the different aspects. So for instance, some children have much more clear vascular changes to the small blood vessels in the muscle. Either those are damaged or they're actually being attacked directly, or sometimes they're actually missing across the muscle. So these features are all fairly easily recognized. I don't think they need specific training, but of course, the people we brought together were experts and very good at recognizing these things. And we went on to validate this score tool in a second study where we also had a cohort of biopsies taken from the upper arm rather than the upper leg, so-called biceps. And that was interesting because we had to first see whether that would make a difference, whether the muscle from the arm was different in the features we wanted to measure. Actually, it worked just as well. And we found, as you might expect, that the more severe the biopsy, the more weak the child, but also certain types of JM, so for instance, identified by their different antibody, had more or less features that were characteristic on their biopsy. And this is that analysis. We also did a really fun thing, which the clinicians between you will probably smile at. We asked them to score a VAS, a line between 0 and 10, as we're all used to doing for nearly all of our patients. Most of the pathologists in the group hadn't done that before. So that was kind of fun, but they got to like it. And I think you'll see in this top right-hand panel that their total score, that's adding up all of the features with appropriate weighting by their advice on the different parts of the 4 domains, compared to their overall assessment of severity, was very highly correlated. So that just made them feel that, yes, indeed, they'd made a score that kind of fits their clinical opinion. It's a nice internal validation, really. And here's the study by the antibodies where those were available. And you'll see that, for instance, the Me Too cases tend to have a very high biopsy score. Interestingly, though, they actually typically get to a better state, a remission state, or certainly an improved state and off steroids quicker than some of the antibody groups, while some of the antibody groups are very heterogeneous across their muscles. So NXP2, TIF1 gamma, and the large group of patients who, at least in 2005, we didn't know what their antibody was. And these are just some images in case you're also in a center that uses biopsies and you're used to looking at these. And this is, of course, a tiny part of the heterogeneity we saw, but all of these biopsies can be scored and can be compared to each other using this tool. And I'll come back to the uses of the tool in a moment. So Claire Deakin is a very talented scientist who joined us at UCL about 10 years ago now. And one of the first things she did was really consider the wealth of data that we have in the JDM network and study, which Andrew mentioned earlier. And this is one of the innovative things she did. She decided to combine clinical data with biopsy score data and antibody data. And we had all 3 on 101 at that time. And she first asked whether the antibody and a combination of that with the biopsy score altered your chances of being on or off treatment over time. And you can see on the next graph how that looks for certain antibodies. And surprisingly, perhaps you're surprised, we were initially, but it's very clear now from other cohorts, not just the UK cohort, the patients with the Me2 antibody, the so-called Bohan and Peter classic cases, they are very like the descriptions in that very early paper 30 or 40 years ago now, actually get off treatment the quickest, and that was significantly different to other antibody groups. But when you take the other antibody groups, so NXP2, TIF, and those without an antibody, the combination of the data, so how high the score was and the MSA, when you combine those in a model, helps you to advise the family how quickly it's likely we could the child could get off treatment. And this is a really important outcome for families, particularly getting off steroids. And in most of my patients, getting off methotrexate would be nice as well. We didn't at that time, within this 101, have enough to then substratify for the other important outcome, who's going to need more than methotrexate and steroids, who's going to rapidly progress or require other drugs like IVIG, cyclophosphamide, these days rituximab. So this study couldn't address that really important question. So I'm delighted to say that actually the score tools has been used in many cohorts. Funnily enough, it's been used in ways that we didn't originally design it for, but I think that's okay. I'm glad that it's useful, and I know some of these colleagues, obviously many of them in pediatric rheumatology, so we sometimes discuss this. And I think it's rather, as has been the case for, well, at least 50 years in the renal community, just having an agreed international tool that genuinely had international buy-in from many experts has helped them decide how to measure those biopsies. And we can now compare these cohorts because we have a standardized tool. So more recently, we've been very interested in trying to understand not only the interferon score. For me, these days, that's just the positive control. Not that it's not important. We're going to do drug trials that may help us to block interferon, and I very much hope they are successful. But if a student is doing an experiment looking at either proteins or genes that are related to the severity or the clinical features, and she or he comes and tells me that they found interferon signature, I say, great, that's your positive control. What else have you found? And actually, this is work published by Meredith Wilkinson over a year ago now that was completely new and for us very exciting. So this was work actually using blood from children with JDM. So all of the children in our cohort study are kind enough to allow us to keep a bit of extra blood when they're having bloods in the clinic. And what Meredith did was sort the cells into different types. So this is just about the monocytes, but we have a lot of data on the other cells as well. These are cells that are very important to the immediate part of your immune system. So they're generally good cells to have. But unfortunately in JM, they're fairly sick cells, and they're sick in the part of the cell that is really the powerhouse, the battery, if you like, the place that the energy is made. And you may have heard of this thing. It's quite often talked about now because it's interesting. It probably comes from very early bacteria billions of years ago. And it's called a mitochondrion. And here is one in a normal, healthy child looking blue. I don't know if you can see my pointer, but in the right hand of your slide at the top, there are 3 blue images. And those are fairly healthy, normal cells. And the blue picks out their mitochondria. But in the JM kids, before they've even started treatment, and in fact, even when they're on treatment, their mitochondria are really extremely unwell. And Meredith showed not only did they look bizarre in the microscope, but they also don't function well. They're biochemically sick. So of course, muscle has to make energy. So we were incredibly excited then that Meredith decided to go back to the muscle. This is from muscle that's stored for 10 or 15 years, a long time in the freezer. It's perfectly good for this experiment. And Meredith and her very able student, Aris, I think, who's not on the call, but who helped with this analysis, have generated some data directly from the muscle taken before treatment started. And this actually shows us something very similar to what we've seen in the blood. So that gives us further hope that the new targets that this suggests, which would actually influence, if you like, the biochemistry of the mitochondria and perhaps return them to a better healthy state, would actually be a suitable target for the muscle as well as the blood immune cells. And the last thing that I think is so important about obtaining and having muscle biopsy tissue is that sometimes we don't know the diagnosis. And through my life of looking after children with JM, actually, there have been new diagnoses that have been popped into our list that didn't exist 20 years ago. So one of those examples has a terribly difficult name to say at 8 o'clock in the evening, but it is immune-mediated necrotizing myopathy. We still consider it under the umbrella of JM, and actually I think it's great that you guys are called Cure JM, because these children needn't be excluded, but we no longer exactly group it with JDM. And the reason for that is partly these children don't have much rash, but they come to hospital very weak with a very high CK blood level, and people know there's something up. And actually their biopsy is really often quite different. And the more normal biopsies your pathologist has seen, the easier he or she will be able to say, hey, this is a different one. This is not JPM. Have you tested this and this and this antibody? So we have 2 antibodies for this disease. I suspect there are more. There are a few of them that don't yet have an antibody, but I think that's just we haven't found it. So it's really important to upskill your pathologists, but they can only do that if they also see normal biopsies, and probably they need to see quite a few. One way to do that is to share images around the world with consent, and we can do that now technologically very easy. So that's one example of where having all of those biopsies already stored when we came to study, this is only 3 cases, this paper, meant that we could go back and see how they were distinguishable from other cases on their biopsy. And then finally, this was actually a patient of mine, 13-year-old young man who came in with a lot of muscle pain, a bit atypical, but a sky-high CK, and had been sent to us as a juvenile myositis, but something wasn't right. He was weak and he had a lot of pain, but he didn't have any other features. He didn't have an antibody. By this time, we were measuring the antibodies at Great Ormond Street. And actually, we got our neurologists involved, and they said, hey, let's stain for a whole set of other things that are not necessarily part of your score tool. And indeed, he had a fairly unusual type of this limb girdle muscular dystrophy. Now, we all know that muscular dystrophy can mimic JDM or vice versa. We sometimes share patients, and so perhaps this isn't that unusual. But I think the very fact that we're already working by luck with the same pathologist who sees our cases and these cases made our kind of multidisciplinary join up for this case fairly quick. And so we reached the correct diagnosis more quickly than if we'd never done biopsies. So I'm going to stop there, conscious of time, although I can't see the clock, by just some really important acknowledgements. So on the left of this slide, you see the fantastic set of investigators who are our lead clinicians around the 17, soon to be 18 hospitals in the UK, nearly all 4 nations. So we've just got Northern Ireland to add. And in fact, the Republic of Ireland is also involved in other ways in this study. Some of their photos are in the top. I couldn't squeeze in every single photo, but really importantly, the patients, parents, and families who don't only allow us to do this work and allow us to keep the data and store samples, but also are very engaged in developing new ideas for all of the research we do. And of course, again, the International Biopsy Working Group, who were just the most fantastic colleagues to work with, and our funders, and you for listening. Thank you very much for your attention.
Thank you, Lucy. We can now open it to questions. So if people have questions, if you could put them into the chat box, and as they pop up, Lucy, if you could read them and address them.
Let's see if I can find the chat. Yeah, I can find the chat. Good. It's always a relief when the technology works, Andrew.
I know.
You know. So please don't be shy. There's never a foolish question. There's no such thing. And if you're wondering, this is Ireland behind me.
There are some questions, Lucy, in the chat.
Good. Oh, good. Hi, Jeff. So actually, Jessica had the
Jessica.
first question.
Mm-hmm.
Do you feel you get the same prognostic information for a needle biopsy as open? Fantastic question. So Jessica will know that we moved to needle about 6 years ago because it was felt by the clinical team to be less invasive, easier to do when the child is really quite sick, and also obviously an open biopsy leaves a couple of stitches or may leave a small scar, which for some people is then unsightly. So the short answer, Jessica, is yes, we believe that as a team, but I'm always for proving that. So on the list of a project to do is that we have started to collate a comparison. Histopathologists who've done this now with us for 20 years, and 1 or 2 of them have been with us throughout that time, have seen both. They use the biopsy tool in the same way they do the stainings. Obviously, we have slightly less tissue available for research, but that's okay. We're now good at doing micro experiments. So I guess what you really want to see is a p-value, and that awaits a good student to do that project. But we will definitely be making a formal comparison. Does that answer your question, Jess? You're welcome to jump in.
Yes, thank you. I look
Thank
forward
you.
to seeing those results.
So we've got here what I call the triple J, the J cubed. Do you know that's what I call you guys? I don't know if you know that. Because we've got Jessica, and Jeff, which is fantastic. I'm very honoured to see you all. So then there's a question from Jeff. Lucy, what do you have to say to those of us in the US where biopsy is not standard of care? Oh, well, what can I say? The short answer, Jeff, which is how I talk to families about it, is that until you start to study the biopsy, you don't know if it will have something useful for the clinicians worldwide. So we were very privileged that actually the foresight of Kevin Murray and Clarissa Pilkington, when I was a new fellow, was to take biopsies and ask that question. Now we feel it really does have prognostic information, albeit a small piece of the jigsaw. It's not instead of doing an MSA or doing an MRI. And in other fields, much more serious sometimes, of course, like oncology or cancer care, I don't know of a parent who would say, I'm not going to consent for my child to have a biopsy because they want to get all the information. Perhaps I'm wrong, perhaps there are some families, but that would be unusual. So if we're sure it has prognostic information, then I think most families are happy to have it. I would say that, of course, it's not compulsory. Of course, it's only with parental consent. And so if the family have a concern about it, we won't do one and we will manage. But we couldn't have done any of this work until we got to 100 or more. We're now up to over 200. The other thing to say is that in my experience, the parents and families of children with JM are incredibly generous people because they also want to help families in the future. And when you have a child who's that sick, that you can still be that altruistic is fantastic. So many say, well, even if you don't use the biopsy because you already know she's got JDM, I'm happy if it helps others. So do you think that would persuade your colleagues or not, Jeff? Maybe not.
Yeah, I know there's a lot of talk going on amongst, the clinicians here, especially the group that meets through TRJM and through CARA about muscle biopsy, and especially, can needle biopsy be a surrogate for open for us? And that's
definitely
easier for us to maybe work with parents.
Yeah. And the other thing, Jeff, I would say, and I don't say this to kind of blow the trumpet of Great Ormond Street, but maybe we won't need to do them in the future. We don't need 1,000. We have 250 odd. We can do that work. And as you know, we're sharing tissue with others around the world. And again, families are very generous to consent to that. Maybe as a community, I'll probably be long gone. In 20 years' time, you'll all come together and say, you know what, we've got enough information, but we now have other biomarkers that we can measure in the blood. And wouldn't that be fantastic? But if you don't do that work, you'll never know if that's the case. So in the future, maybe they won't be necessary. Maybe I'll keep going, Andrew, because I'm conscious of time. In a way, the same question, I think, from Jess Turnier. Did we get comparative information on needle biopsies? Suzanne says, this is something we should talk about. Great, fantastic. Any newer immunostains we should do a muscle biopsy for other? Yes. So one of the things I think Jessica Turnier asked, should we be doing new immunostains for candidate tissue-based biomarkers? There's a few things there, Jessica. So number one, MDA5 positive cases still have a biopsy. And around the country, my colleagues often send, if they've done a biopsy, a piece of tissue, a block of tissue to our histopathologist because their pathologist has said it's normal. And that's because they haven't done an MHC class 1 stain. Very simple. That will still be up, remarkably enough, only slightly. Apart from that, you'll see almost no inflammation. As you know, they're generally not very weak. But we wrote that up, a very early paper by Lisa McCann. So that's not new. That's bog standard. Every lab in the world can do it. I would argue yes on the mitochondrial work. It's well worth talking to the mitochondrial experts in your centre. Most large children's hospital will have one. And sometimes we add those in. But the only reason I'm hesitating is we built the score tool with a set of rules. And the number one rule is that we would build something that was going to be cheap to do all over the world. So it doesn't have anything very fancy in it. And I sort of hold that up as a caveat immediately. It literally has stains that any histopathology lab is likely to be able to do. So therefore, we haven't got data on all 250 on a whole range of other, if you like, more sophisticated markers. I don't know if Mary would like to comment about mitochondrial staining or analyses, maybe in the future, Mary.
Yeah, we are running a little bit behind time, but I think that really just exemplifies how much interest there is in this, not only your presentation, but this whole area of biopsy samples. As Jeff pointed out, it certainly is not standard of care in the US. So addressing how we should change and potentially going to needle biopsies is most stimulating.